Hello, students. Welcome to this class on neurobiology or behavioral neuroscience, depending on if you're looking at Canvas, the course catalog, or class search. Sometimes it's called neurobiology, sometimes it's called behavioral neuroscience, but this is the Bachelor's of Applied Science in Behavioral Health program, class 350. So we are going to be talking about this first chapter on the introduction to biological psychology. So this is your textbook that we are working from. It is the current edition is called the revised edition. It was published in August of 2024, and this is what we'll be working off of. You can download this in EPUB or PDF format, and this is what the first chapter would look like if you're reading it on the web. So neurobiology is a fascinating field, one of the fields I'm most interested in. If I was to go and do another degree on top of my PhD in positive developmental psychology, it would probably be in neurobiology. But rather than that, I will just connect with people who are doing this research and work with them on related projects rather than trying to go through that whole process again, which I think would be a lot easier the second time. Anyway, neurobiology is looking at the biological factors, specifically in our brain, but we'll talk about other body systems too, like the digestive system, other aspects of our body, and how the brain, nervous systems, our hormones, our genes, how these influence cognition, emotions, motivations, memories, actions. This specific textbook does a pretty good job at looking at the behavioral health side, has a lot more focus on things like medicines, disorders, and the neural substrates, which is basically the term for the neural basis, so neural substrates of where these disorders might come from or some connections that we see. A lot of these are correlations. We do have some causal data from animals, and we'll talk about that today in a little bit of detail. So this is, what is your brain doing? Why is it related to the things that we see from behavior, from things people are feeling, from how you think about things, how do you process memories, all sorts of things. It's a really key component of this whole process. And so we really do want to help you understand and know about these processes. You might not come out of this class feeling like you want to do research in neurobiology, but hopefully you find a lot of really interesting things that can help you understand when you're working with people in the field. So with this, let's get a couple interesting facts. Humans in general, on average, have about 86 billion neurons in our brain specifically. And with that, inside the brain, we have about 300 million neuron connections per cubic millimeter. So that's a very small space. So if you have a ruler around, grab that, look at a millimeter, imagine that as a cube. About the size of a ballpoint pen, you have 300 million connections. So there, it is a very complex system of connections, and we'll try to break it down in a lot of simplicity. But hopefully the simple models and examples that we see lets you see that, yeah, there is a big complex system going on for everything that we're involved with. So the brain is about 1,200 cubic centimeters, about 3 pounds. This means we have about 60 trillion synaptic connections in the brain. And so there's essentially infinite possible activation patterns. Everything that you experience kind of runs through those kind of processes, the things that you learn, so your thoughts, your movements, your perceptions, your own sense of self. All of this emerges from those electrical chemical signals in those specific networks, in the specific patterns in your brain. So sometimes we'll actually use Inside Out as a little example. It's actually not a bad example of some of the processes that your brain goes through. Obviously, it's a fun movie. It has a story. There's not actually little characters in your brain, but they definitely talk to some people who knew about the brain in and how emotions work in creating that movie. So these pictures here, this one I'm hoping helps you see like some of the little areas of the brain. So we've got some of them towards the frontal lobe, some towards the occipital lobe, and each of these is kind of identifying an area where there's a lot of activity happening and it's connected to other activity areas. So when you are doing any kind of process, this is the kind of activity level that we're looking at in your brain. It's very complex. We'll talk about how we get these types of pictures later in this chapter when we're talking about research methods. But this is a basically a printout of all of the axons, or not all, because again, there are so many trillions, but it's mapping some of those axon pathways. So you can think of each of these lines as an example of a lot of axons are running around that area. And so those axons are the main delivery mechanism, the message highway from the neurons. So it is a very complex, very connected organism. So why do we study it? Why do we look at the brain specifically? Well, the main point of this class is to get a lot of these key concepts that are really relevant to the behavioral health field. Again, the book that I picked for this class is because it is a little bit more focused on behavioral health. So some of the chapters are very, very focused on whether it be medicine, whether it be disorders. And in this, I'm hoping that through this course, you get a lot more understanding of some of the neurobiology basics or connections that'll really help you work with the clients you're planning to work with. So if you understand how their brains are working at least a little bit more clearly, you might be able to help them understand that. I know for me, going through all of my classes and learning about neurobiology, it's helped me learn how to use my memory better, to do things like studying classes, how to take breaks, to work around how my brain and how me as a person with this brain works. So these are things that you can learn yourself. These are things that you can learn to work with clients. You might not have a super deep interest in neurobiology, or it might be really fascinating to you. Either way, the main idea is that each of the people that you interact with have this complex brain happening all the time. So understanding how that's working is going to help you with working with these people. So a lot of the focus here is going to be on language. So you could think of it kind of as a language immersion class. So you're learning specific definitions, specific parts of anatomy in the brain, but really we'll focus a lot on the breadth of what the brain is doing for any given process, for any given disorders. And we get a little more depth in your assignments, so you can dive a little more depth into those assignments. But with this, a lot of these first couple chapters are a basics on how is the brain connected. So what are the brain areas? What do they basically do? And then we'll get into how are those involved in when you use this specific type of medicine? How are those involved when you have this specific diagnosis? These kinds of things. What happens when you have trauma or stress? So we'll talk about that. So think of this kind of as immersing yourself really briefly into learning about the brain, learning about neurobiology, learning about the kinds of language that we use so that when you see it in a paper, when you see it in a report, when you see it on someone's diagnosis, something about their brain, you can have a lot clearer picture of what's going on. So this is an example here, again, of some of the connections in our brain. This is either looking for the from the back or the front. I think this is looking from the front. And this is the corpus callosum, which is one of the main connecting highways in the middle of our brain with a bunch of axons going from both sides. So one quick thing, hopefully I've flipped the camera correctly, but I am right now moving my left hand, waving it around in the air. And that is being controlled by this right side of my brain over there, right about there. So that message goes through this corpus callosum and to my brainstem, down to the motor neurons in my hand to wave my hand at the camera. So it is a very complex system, and we'll break it down to make it a lot more simple. Some of the basic functions and structures of the brain really are simple. The way that the neurons work and how they connect and start to communicate with each other, link up into those patterns, is really pretty simple. So once you understand that, I think a lot of these things will be a lot easier to understand. So with that, what are some of the different areas of biological psychology? There's a bunch of different areas that we will connect to throughout the course. I've already mentioned psychopharmacology. So this is specifically looking at drugs and substances affecting the brain and behavior. That could be illicit substances. That can also be substances that are prescribed. Neuropsychology looks mostly at brain function and behavior. A lot of focus is going to be on things like brain damage or traumatic brain injury, any kind of dysfunction. So something like Alzheimer's, and how that affects our cognition. Uh, Here we have psychophysiology, which looks at other physical processes as well as psychology, the brain, but also looking at things like the heart rate, EEGs, other physiological processes in looking at the brain, body, behavior in general. Psychoneuroimmunology looks at the nervous system, the immune system, how we can have, say, stress hormones talk to our body, how things like your immune system are interacting with your brain. So these will again come up throughout the course. So if you go further into looking in neurobiology, you would have specific courses in each of these domains, kind of like when you were taking maybe a general class. Every given chapter is kind of a domain or area of the field. So evolutionary psychology will come up a lot, especially since a lot of the research that we do is using animals that are closer or further related to us. And so evolutionary psychology looks at how evolutionary processes like natural selection might have shaped some of our human behaviors, as well as the ways our brains are formed, how they develop over our lifespan, this kind of thing. And the behavioral genetics is pretty similar. It looks at those genetic factors, those contributions from our genes, looking at differences between behavior, cognition, personality, how much or how little is affected by genetics. And that's a big key piece, because if there's something that is entirely genetic, then there's not a lot of things that we might be able to do to interact with that unless it's something like medicine. But if there's something that's a lot less genetically tied, maybe there's more we could intervene there. So these are important things to learn. Cognitive neuroscience looks at kind of what's going on behind the scenes in your brain. So if you think of those inside out characters, I kind of think of them as the cognitive neuroscience piece. They are the ones doing like the attention, like pointing. The main character of the movie is Riley. If you haven't seen it, feel free to go watch the first or second one to get an idea of these examples. I'll probably bring up a lot. Memory, language, decision making. So how that happens, those kinds of processes are what cognitive neuroscience looks like. And then comparative psychology really looks at comparing our brain processes to non-human animals and see what are the similarities, what are the differences, what are the kind of evolutionary patterns that shape those behavior and mental processes. So you might compare how a rat responds to stress and as a human responds to stress. And as we'll talk about, there's a lot more in-depth investigation that you can do with, say, that rat, with the types of methods you can do to investigate what their brain is doing that you can't with humans. So this is a really important piece. And a lot of times, any given piece of research will be using multiple of these divisions of biological psychology. So it's a very interdisciplinary field. Looking at how the brain works is not just the area of psychology, it's also the area of neuroscience, it's also biologists, computer scientists, philosophers, medicine, engineering. I was just looking at a paper the other day that was from chemists as well. There are a lot of people that are doing work that is really related to neurobiology. So it's a very interdisciplinary field. With that being said, this is a cool example that's really directly connected to my research. A lot of my research is in heroism, prosocial behavior, these types of things. And so one of the people that I have collaborated with, met at conferences, talked about heroism with, is Stephanie Preston. And this is one of her papers on the origins of altruism in offspring care. So she's looking at, well, this specific study was looking at, do rats take care of other rats that are not part of their family line, like completely genetic, different line? Will they take care of them at risk to themselves, like give food that will make them have less food? Like how big of a risk will they take? And these rats will take altruistic action. So where does that come from? And so the idea here was posited that maybe this is also because this is rooted in instincts, or it might be choice, right? So that's kind of the idea, is this comparative psychology, looking at the behavior of these similar mammals and how their brains are working. And we'll see a lot of other cool research with mammals as well. This is another really good connection that hopefully for you in behavioral health, you've thought of some of these, you might even know this process, but here's a short video to show you another connection between these interdisciplines that are looking at how the brain works and how it can be related to treatment.
Naloxone, also known by the brand name Narcan, is a medication that can be used to treat opioid overdose and substantially reduce the risk of death associated with opioid toxicity. The drug is a competitive antagonist at opioid receptors, which means that it works by binding to the same receptors that opioid drugs bind to, But instead of activating those receptors like opioid drugs, naloxone blocks activity at them. Naloxone also causes opioid drugs that are already attached to the receptor to be displaced. This combination of receptor blockade and removal of active opioid drugs makes naloxone capable of reversing an opioid overdose and reducing the risk of complications associated with one. Naloxone is commonly administered by injection or nasal spray, but intranasal administration has become a preferred method due to its ease of use and safety. The drug typically begins working within a few minutes, with the exact time to onset being dependent on the route of administration. The effectiveness of naloxone can vary due to a number of factors, including the potency of the opioid in the patient's system and the dose of opioids they administered. Repeated doses of naloxone are sometimes used to keep the patient from re-experiencing effects of opioids still present in their system after naloxone begins to wear off. When naloxone is administered to someone with opioids in their system, it can cause acute withdrawal symptoms due to the sudden cessation of activity at opioid receptors. While there is some risk associated with complications of acute withdrawal, it is considered low, especially compared to the risk of complications linked to opioid overdose. Naloxone administered to someone who has not used opioids recently typically has little to no effect.
So hopefully you see a bunch of the connections here between the fields that you are interested in, as well as all these disciplines that are looking at neurobiology. So hopefully the example of naloxone or heroism and prosocial behavior can help you see all of the different ways that looking at brain and behavior, the way that we are working in our neurons, really all the way down to that molecular level of the neurons is really important for all of the things that we do. Another big thing that we'll look at in this class are the healing of the brain. One of the big reasons that we study the brain in so much depth is because there's a lot of ways that we can help people with healing the brain. So one of the big ones, especially one of the ones that's really related to a variety of therapies, is increasing plasticity. So you may have covered plasticity in hopefully a general class or maybe a lifespan class. But plasticity is essentially the idea that your brain can make changes. Even late into life, that your brain is capable of making new connections, making new patterns and new networks. And we'll talk a lot more about that when we talk about the structure and function of neurons in the next couple chapters. But the basic idea is that at every point in life, whether you are very young or very old, your brain is relatively plastic. It can make changes. You can teach an old dog new tricks. And so that is a lot of what therapy is trying to do, is at the neural level, you are literally making neurons talk with each other in a little bit different way than they had in the past. And that can lead to a lot of positive effects. As we talk about things like stress and trauma, you'll see those things can have a lot of negative effects, plasticity in that way. But with that, another thing that we are looking at a lot is regrowing neurons. So stem cells, and we'll go into a lot more depth on this, but stem cells become neurons. So neurons themselves don't divide into new neurons. That would be pretty neat, but that is not how it works. A stem cell can divide to another stem cell, and then one of those stem cells can become a neuron. So there are a couple places in the human brain that this happens. It seems like there's more places in, say, rat brains that this happens. So we're trying to figure out if there are ways that we can increase the production of those neurons to really make a difference. As of now, it doesn't seem like there is much measurable difference in the kind of the small number of new neurons that we have. New drugs as well. How do those affect the nervous system? As we'll see as we get to the area on psychopharmacology, the neurotransmitters that they affect do a ton of things in our brain. And so having drugs that are a lot more targeted, that are a lot more specific on what they can do, can be very helpful, can reduce side effects. Other things like transcranial magnetic stimulation. We'll actually watch a video on this in just a second, but this is a way that we can interact with neurons in the brain, making them more or less active for a variety of reasons. So we'll watch an example of transcranial magnetic stimulation, or TMS, being used for depression specifically. There's also other methods of deep brain stimulation that we can do with other technologies. There's also a lot of ways that we can connect brains to computers. So there are ways that we can help teach people with biofeedback. You probably heard of biofeedback already, but looking at a screen of what is your brain doing, what's your heart rate doing, what's your blood pressure doing, what is your sweat level doing, and seeing an actual interface of these are some readouts of what your brain is doing. You're more stressed, you're more calm, can really help in a process of, say, learning how to breathe and be mindful in any given situation. But a lot of this is to help heal people's brains in a variety of ways, help their brains work in a way that maybe they don't have those triggers from trauma anymore, that they don't have the addiction anymore. So, or that they have skills that they know, that they have plasticity to work around some of those triggers for whether it be addiction, trauma, whatever it might be. And we'll get into some more specifics. But let's watch a couple videos. So, any of these videos, I have had times in various classes where I record the video along with my slides. And in some cases, these videos have either like had to be deleted from the video that I post on YouTube, or it's muted because they say it's copyright material. And this is fair use of it. So if we actually went to court, they'd be okay. But main point is if the video for some reason is not working, just go to the link in your slides. So if it's muted or it's not actually there when I say, hey, we're going to watch this video, and then it doesn't pop up, just go and click on it on your own. So I'll play it as if I assume that it will be working. And we'll find out later when I put it up onto YouTube if it actually did. Sometimes they like to delete or make me cut out things. So let's watch this first one on depression being using TMS or transcranial magnetic stimulation as a treatment for depression.
Treatment-resistant depression is generally defined as depression that hasn't gotten better with trying 2 antidepressant medications. We know that after a person has tried 2 medications and has not improved, their chances of responding to a 3rd or 4th medication are much lower. We're excited to offer transcranial magnetic stimulation, or TMS for short. This is a non-invasive treatment that uses gentle pulses of magnetic energy to stimulate nerve cells in the brain. It works very differently than standard psychiatric medications, and it can work even when medications haven't been effective. When a patient comes in for TMS, they sit in a chair that looks a lot like a dentist's office chair, and we place a magnetic coil over a specific area of their brain. That magnetic coil generates gentle pulses of magnetic energy that will stimulate the nerve cells in that region of the brain. So when these gentle magnetic pulses reach the surface of the brain, they cause the nerve cells in the brain to fire and send a signal. What that does will depend on what part of the brain you're stimulating. For example, when we place the magnetic coil over the motor area of the brain and we send one of those pulses, we can send a signal from that nerve cell all the way down a patient's spinal cord, down the nerves in their arm, and cause movement in their hand. For depression, we're treating a different area of the brain that's involved in regulating attention, thinking, and behavior. TMS is also helping to rewire the connections between that area of the brain and deeper regions in the brain called the limbic system that are responsible for regulating mood. The treatment feels like a firm tapping on the head. Patients are awake and alert during treatment. They don't need any sedation or anesthesia. There's no recovery time. Patients can return to their normal activity immediately after treatment. The most common side effects are some discomfort at the treatment area and potentially mild headache after treatment.
How do you feel, Kate? Good.
TMS treatment usually lasts for about 20 minutes, and it takes place 5 days per week for about 6 weeks. The benefits can be long-lasting. For some people, the benefits last for a year or potentially much longer. But I will say that unfortunately, it's not a permanent cure. We just don't have a cure for depression yet, but TMS is a wonderful, long-lasting option.
Before jumping to the next one, I just want to make a couple mentions here. So in that video of TMS for depression, one of the main things that they talked about is that they're doing this every day of the week for about 6 weeks. And one of the ideas is to retrain connections in the brain. That's exactly what we're talking about in plasticity here. So they're trying to interfere with a pattern that the brain is using. So the neural pattern that it's defaulting to, or they're making other areas active that aren't usually as active. And this helps by making those neurons active, trying to send out messages, and that means it now becomes part of a new network. That's the goal there, is to over time help the person's brain and help them train themselves and train their brain. So overall, just train themselves to be able to run different patterns in the brain. So you're not defaulting to, say, those um, ruminations for depression, for example, or you're able to switch to a different network and think about something that you learned from your cognitive behavioral therapy session, right? So you can switch attention a little bit better. So that's plasticity. And so that TMS is trying to target like a specific area of the brain and try to activate or deactivate or interrupt the activity of those specific neurons. Pretty neat. So let's look at another one at the brain connecting to a prosthetic specifically as another example of healing people.
That brings you back to a sense of normalcy. It's like you have a hand again.
Advanced prosthetic hands on the market can do lots of things.
These are beautiful prosthetics with incredible capabilities, but we've had no way to control them.
To get that kind of control, you really have to go to the nerves.
The problem with most of the technologies we have is that the signals are really tiny. You have tiny little peripheral nerve signals, and you have noise in those signals that's about the same size. So when you try to hear what a peripheral nerve is saying, you actually can't hear it. We designed a way to connect up with the peripheral nerves with a piece of muscle. And then what happens is when a tiny little peripheral nerve signal comes down the nerve, it goes into the muscle and it becomes a huge muscle signal.
We've now seen.
So I'll interject here really quickly and then we'll go on to the rest of the video. So what they're saying there is with our current technology, we have peripheral nerves all throughout our body, motor neurons, right, that are sending messages that are sending messages to other neurons that eventually end at the motor neuron. And what they're saying is our technology does a really poor job at reading that message. The subtle differences between a neuron active and inactive are so small that with our current technology, it's really hard to read that. So what they did is they put a muscle cell or probably a cluster of cells, and that the movement or the contraction of those muscles, our technology can read that a lot better, and that is what they're doing. So they're using a combination of looking for that signal and also looking for that muscle contracting. So using the body, using our biological systems in a way that our technology can work with it.
To my knowledge, the largest voltage recorded from a nerve compared to all previous results. That makes these signals big enough that we can record them and interpret them for controlling a prosthetic hand.
Brought back into my mind the thought of, well, if I had something like this, I could actually be out working without risking hurting myself.
I think it's a really good step into the future. It's a good way to move forward for not only me, but for other people.
You can make a prosthetic hand do a lot of things, but that doesn't mean that the person is intuitively controlling it. So the difference is the person just thinks about moving. This worked on the very first time we tried it. So now we can access signals associated with individual thumb movement, multi-degree of freedom thumb movement, individuated fingers, and this opens up a whole new world for people who are upper limb prosthesis users.
I do wish they had held on his face there smiling for a second, because I think that's a big part of all of this. When we're looking at behavioral health, when we're looking at how to help people, that's one of the big things that we want to see is that kind of encouraging behavior, that those smiles, people making connections, people making changes. So I do wish that they had held on that for a little bit longer in the video, but that's okay. Hopefully you can see how this can be related to a lot of people that you might interact with in the field. Also, I was thinking as we were watching that video of a completely unrelated topic of, hey, you know, this would be a step towards making giant mech suits like Pacific Rim, where we can control the mech suit rather than a robotic arm, a big robotic arm and a big robot. So not really related to the class, but maybe that connection will help you as well as you think about this. So there's another video here that I won't watch. If you want to watch, it is a really good one. It is actually implanting electrodes into a person's brain to see if we can use a prosthetic and a touch sensation on that prosthetic to send a signal to the person's brain that they are having a touch. That kind of thing, the putting electrodes into people's brains for research essentially doesn't happen unless a person is already getting brain surgery for something. So one, it's very rare, and two, it does show the person's brain, and so obviously blood in the video as well. So if you're interested, it is a good one, but I won't watch it right now in the class. All right, another big application of biological psychology is to try and encourage social policies that are good for our brains. So, humans, even with neurotypical people or neurodiverse people, our brains all work in fairly similar as well as dissimilar in some ways, dissimilar in similar ways. So with that, we know how humans thrive. Like we're getting a lot better understanding of what makes people's brains happy, what makes us happy, what leads us to be productive and feel good about ourselves and learn well, remember things, so with that, we can make social policies based on how humans actually work. So a really good example of that is high school start times. So when in person, if we have this discussion in person, I'll usually ask, well, who had the earliest high school start time? Some students had to get to school, like actually be there at the school building in between 6 and 7. So I think the earliest that they had, say like a pre like before school band class was 6:10 was when it started. And so they had to be there like at 5:55 or something like that, which is absurd for a high school student. That's not how brains work. You need more sleep. So, high school start times are a really good example of social policy that can help young people learn. Another good example is work week. How many hours should you be working? How many breaks should you be taking? Well, we know a lot about the brain, and generally, we need a lot more breaks than we have in day-to-day work. Usually in my classes, I try to do a better job of this, of taking breaks during class. Any given person should be anywhere between 20 and 50 minutes. You should take a break. If you have a shorter time, you should take a long, or if you have a shorter work span, you should take a shorter break. Longer work time, closer to that 45 mark, take a little bit longer break, up to even 15 minutes. And that we'll talk about that again when we get to learning, attention, memory, things like that. Addressing drug addiction. This is very relevant for a lot of you in the class. Is what kind of social policies actually work to help people with drug addiction? And you've learned a lot of them in other classes. A lot of those are directly related to how the brain works with a person with addiction. Also, serving the mental health needs of prison populations. So, how should we help these people who have mental health care needs that are in the prison population? And generally, they need a lot more support than we are giving them. So, based on our brains, can really help us know what we can do to help other humans, to make us all thrive, make society better just in general. So a little bit more sad meme here is over here. We've got someone thinking about, well, should we pay adults a living wage to combat the labor shortage? Or should we change labor laws to allow children to work longer hours for poverty wages? Well, the better option would be the living wage and shorter work hours for everybody, not just the children, but the adults as well would be better for their brains. Another big area that biological psychology is connected to is AI research. So I was just reading in a paper about AI the other day, about how they use rewards and punishments, so reinforcements, to reward AI for doing closer to or to do the thing that they are programming it to do. That idea came from biological psychology research in learning and how our brain works. So giving an AI reinforcement, programming it to say, this was good, do more of that, came from biological psychology. So modeling computer architecture as well, our own human brain neural networks have really helped to improve computer speeds. So we're still learning about how connected our brain is, but from what we know now, there are multiple layers of connection. So things like that help AI, people modeling AI to do things, whether it's large language model like ChatGPT or an image generator, So a lot of these things are really helpful to look at our actual brains and try to model them. So with that being said, quick reminder, do review the AI policy in the syllabus and on Canvas, the material on what the expectations are in this class for the use and citation of any use of AI. Now we get to one of the things that I think is really interesting. So your book has a briefer section on this in research methods, and I want to dive into a lot more depth so you know how we find out this information before we dive into the information. So we'll see a lot about neurons, a lot about specific areas of the brain, and I'll mention studies. We'll go in a little more depth here about how do we do those studies. A lot of research now, especially for comparative psychology, is done with rats, mice, and birds. Non-human primates, such as chimps, used to be used a little more commonly, but that is very, very rare now. Most often, we're using rats. Sometimes, depending on what it is that we're looking at, we might be using mice or birds. We'll also look at things like fruit flies. There's a variety of animals that you'll read about throughout your book about ways that we've learned about brains and behavior. So with this, we do have, hopefully you've had research methods in a other class, learned a little bit about social science research methods and some about the ethics, such as IRBs. We have ethical rules for research with animals. So the kinds of processes are really similar to IRBs with humans. These You will propose the type of study that you want to do with the animal, let them know the benefit. What are you getting from this? What is the cost to the animal? If you are putting an electrode into the brain, what is the cost? What are the risks of that kind of thing? Also, one of the big keys is to restrict pain and stress to the animal. You basically only use any kind of pain or stress to the animal if you can't do any other methods. So essentially encouraging creativity in methods so you don't have to harm the animals in any way. So there are a lot of other ways to minimize discomfort to animals. One of the big things is making sure they live in a healthy, happy place so that there are not diseases, so that they have food, so that they have water, so they have clean cages, these kinds of things. I was able to work with in undergrad actually with rats and train them in behavioral methods. So no kind of neurology for the implanting electrodes, but we taught the rats to do things like obstacle courses. It was pretty neat, giving them reinforcements for those things. But we had a lot of very strict rules and timelines of how frequently they had to be fed, how much we had to weigh them and keep track of that. So if audited, we were abiding by the rules to make sure that these animals were healthy and happy and things like that. One of those rats, we actually got to, well, we took it as a pet for a short time, then we moved to an apartment that wouldn't let it. And so our lab partner actually was able to take the rat and keep it as a pet for, I think it lived for another 5 years after that, which is pretty impressive for a rat. Their average lifespan, I believe, is like 3 years. So it was pretty neat to be able to go visit it every once in a while. And see that rat that we trained again. But there are a lot of ethical rules for research with animals. That being said, this does not negate the criticisms that animal rights activists have towards this animal research. So animal rights activism has really improved the treatment of animals for research. And I'm sure there are a lot of other creative ways that we can do this kind of research to even further limit the harm to animals. I have this picture up that they've saved more lives than 911. This was actually up in the lab that I worked with the rats as well. And in a lot of ways, it's true. There are a lot of things that we do to these animals that we would never do to a human. You'll see some of those examples in a couple minutes, actually, in these slides and in this chapter, and in some videos that I'll share. So that being said, it is definitely a relevant and active debate. You might have very different ideas and attitudes than I do about animal research and what is appropriate, what is ethical, and that's an important thing to discuss. The more that we can think of ways to treat not only our fellow humans, but also fellow creatures on the planet, fellow animals, humans are an animal as well, will improve the field in general. So that being said, my own personal attitude here is that it is a very important thing. That being said, it doesn't mean that I think all of the limits that we have are enough. So I definitely still encourage animal rights activists to try and help us find other better ways to do this kind of research with animals to continue improving and not harming the kinds of animals that we share this planet with. All right, so with this, one of the big things that we're looking at with any kind of research in the brain is converging evidence. And this is the case for all kinds of areas of research, is that you want converging evidence to show that every different field, every discipline looking at an idea is coming to the same conclusion. That's what converging evidence is. So you have multiple studies that are coming to the same conclusion. You use multiple methods to come to the same conclusion, multiple fields coming to the same conclusion, and they're all converging on the same answer. So that's what you want to see. And that's really important, especially in a field like neuropsychology or psychology in general, because there can be a lot of variables that we might not be able to control for. So the more that we have other evidence showing that this answer that we're putting up there is the case, the better. So this image has an example of the theory of evolution. And so hopefully you know from other classes and research methods that theory is like the biggest top thing that we look at. The theory of evolution is like, this is the best explanation for everything we understand about evolution. So what we have there is things like the fossil record. We have experimentation. So things like fruit flies or other really quickly reproducing animals. We've seen evolution in these kinds of experiments. We can see anatomical vestiges, so looking at the fossil record and seeing things in animals that maybe shouldn't be there, like the ancient stumps in snakes where their legs used to be, this kind of thing. Looking at similarities in genes. All of these different fields that found these results in different ways all converge to say, yep, the theory of evolution is the best way to explain that. And that's what we're looking for in the brain. We can't always do what might be the most efficient way to figure out something in the brain, either because it's unethical, we don't have the technology at the time, whatever it might be. So using a multitude of methods, a multitude of fields to figure out this is what this brain area does, this is how it's related to X, Y, and Z, that's what we want. We want to converge on that evidence. So, early research methods in the brain really looked at the behavior of people with brain damage. So, someone that had a serious injury on their head and was still alive, seeing how their behavior changed, for example. You've probably heard it if you've had any other psych classes about Phineas Gage, who had a railroad tie. This is actually a simulation of probably the areas in his brain that would have been damaged by that railroad tie going through his head. So a lot of his frontal lobe and a lot of the connections to some of the thalamus were and other inner, so basically right in the middle of the brain areas were damaged as well, some of the connections between those. Also looking at Paul Broca, the Broca's area for speech, so damage, and then looking, oftentimes this might be done in an autopsy after death. So really people who just happen to have brain damage, then seeing how that was related to their behavior. That was the way we could do it up until we had better technology to get better measurements. More recently, we've had some other invasive physiological methods for studying the brain. One is brain lesioning, and this is intentional injury for parts of the brain. So for a rat, you might cut out a specific area or cut connections between specific axons to see how that changes the rat's behavior. Does it make it more afraid, less afraid? Is that a part that actually is involved with fear? Does it now not have a memory? Is it creating a memory, not able to recall? Social behavior change. We can do this more recently in temporary ways. So we can lesion the brain or intentionally injure the brain by freezing or using anesthetic to shut down specific areas of the brain. And so that's used a lot more because it is reversible. So again, getting more creative with the animal kind of research so that it can be undone. So that being said, it's still definitely risky. Brain lesioning is oftentimes a higher risk thing. Another thing that we could do is implant recording electrodes. So we can read with electrodes individual neuron activity. So we can see, does this neuron have a part in, say, this memory path? Is this neuron part of making an arm move or a tail move, right? Things like this. So you can record on electrodes. You can also implant electrodes that activate a specific area of the brain. They're still not as specific as activating like a single neuron, but a couple neurons. In rats, they're specific enough to activate a specific neuron. But in humans, the recording electrodes and that kind of invasive methodology doesn't happen. So essentially the only time that we get electrodes implanted into our brain, whether for recording or for activation, is if someone is already planning on an operation that opens up the skull. So any IRB is going to say, absolutely not, you cannot open the skull just for research purposes. If you have a patient who's also getting brain surgery for this thing, maybe tumor, something like that. If they're already getting a surgery, that would be the kind of patient who you might suggest as a researcher, be like, hey, would you be willing to try this new method kind of thing, and get an electrode implanted or that kind of thing. So IRB isn't going to approve of that for humans. One thing that we use a lot is called an EEG or electroencephalograph. So this looks at the electrical activity through the skull and the skin. So you can get a little bit of a sense of what the brain underneath is doing through these sensors on the skull. So when a lot of neurons, let's say you've got a lot of neurons active in this prefrontal cortex of the person right here, so right here at the front of their brain, these can detect that small amount of electrical voltage. So it's not very specific for something in the very, say, middle of your brain. With a more complex cap like this, where you've got a lot of sensors, you can get some idea, but it's not super specific the deeper into the brain it's getting. So that spatial resolution of pinpointing where this is happening, it's really low. It's not very good for EEG. It doesn't give you a sense of exactly where this is happening. So that spatial resolution is low. You do have higher density caps, these things with like 256 electrodes that are a little better at it, but it's still you've still got to kind of triangulate between these and look at that signal. There's a lot of variables. There's a lot of substances in your brain, not just the brain cells, but also cavities filled with fluid as well that you've got to go through. So it is possible to get kind of an idea of where this is happening, the spatial resolution. But EEGs are really, really great at temporal resolution. So looking at when something is happening. So, oh, their brain is active right now, to the millisecond, getting exactly right now, their brain is active there. So that's what an EEG is really, really great at, is getting that temporal resolution, getting that millisecond precision of exactly when some part of the brain is active, or when we have these sensors detect activity, then these sensors detect activity. That's a really useful thing for EEGs. Another really useful thing about EEGs is we can use them in a wide variety of contexts, at least now. When they first came out, it was a little more cumbersome, but now they are very simple to use. You can put them on babies who are moving around. There's basically a wire trailing or even a wireless one. You can put them on athletes. There's been EEG studies of people doing climbing or gymnastics or physical therapy exercises or babies crawling around a room playing with toys. So EEGs are really, really useful. They are also tend to be fairly cheap to set up and analyze as opposed to some of the other methods that we will talk about. So they are a highly used one, even if they're not really great for, again, that spatial resolution. They do a really great job at that temporal resolution, pinpointing exactly when something is active. So, and again, they're really flexible to be used in a lot of different scenarios. A similar type of method is called the magnetocephalograph, and it's MEG or MEG. So it's really similar to an EEG, but it's using magnetic fields instead of electrical fields. That being said, it is a lot better at spatial resolution, detecting where something is happening, and the temporal resolution is also better, but it's really expensive. Currently, this is what the model looks like. Not only do you need this whole machine, but you also need a shielded room. So generally, these are going to be in a research station or a hospital where you have something in essentially a Faraday cage where you have controlled magnetic fields because the neural activity or the magnetic fields generated by your brain are really, really weak. It's detectable with a machine like this. And maybe in the future, we'll have Star Trek technology where we can just scan with a handheld thing. That'll be awesome. Right now, this is where we are with an MEG machine. So really similar in looking at when and where activity is happening, and it is better than EEG in that spatial resolution or exactly where in the brain is active. But weakness, you can't put this on a baby's head and have them crawl around a room. So it's not used as much. So PET or positive emission tomography scans look at brain activation, not structure. So we see the brain activation, we kind of see some structures or some areas, but the idea behind these are to look at brain activation. So what it's actually doing is detecting a radioactive substance in a participant's blood. So you put that radioactive substance into the bloodstream, and then a few minutes later, you ask them to do a specific task, whether it's remembering something, playing a game, holding attention, whatever it is that you're doing, and then you look at which area of the brain is getting more blood. Just like with an injury, you get sent more blood. When you are using a specific area of the brain, it's getting more nutrients, more building blocks, the things it needs to do its job through the blood, like oxygen, for example. So the more blood, the more activity. So you're looking for the blood, you're getting more activity. So it is rare in research because you do need specialized radioactivity training. So it is a really useful one for looking at more specific areas of brain activation, but it's not used as heavily as something like EEG is. One that's used a lot is MRIs because their cost has declined substantially. They are still very expensive. So it's not that every single college lab is going to have an MRI machine to let you do research on, but they are a lot less expensive than they used to be. So these are give a very high resolution picture of brain structure. So they're, again, going back to that other slide here. So that spatial resolution. So it's giving you a really good map of the brain in general. So brain structure with structural MRI or SMRI is looking at the brain structure. Brain function, or which areas are functioning, is fMRI. And so with this, you can get a good picture with an MRI if you've ever used one, if you've had an injury that had to use one, you can get a really clear picture of the different kind of tissues, whether it's white matter, gray matter, spinal fluid, bone, blood, tissue. You can get a really, really clear picture with this. A problem here, you've got to sit still. It's very loud. It is still fairly expensive to do an MRI. And so you can't, again, let's imagine a baby going around a room. You don't have an MRI cap that a baby can wear and get an MRI as they're doing some activity. That being said, there are some really creative ways that we can, say, have a older person than an infant. It's not going to work for an infant, but a teenager could maybe sit in an MRI machine, watch a screen, push buttons, do some activity, try to memorize a list of numbers, repeat them back. There are a lot of really creative things that you can do with an MRI machine, even while you have to be still. So another example, a more specific type of MRI is called a diffusion tensor imaging. It makes this really pretty image, really cool. Essentially what it's looking for specifically is water molecules. So it's looking for the difference, just like we can see the white matter, the gray matter, the bone. This is looking specifically for water molecules. So they move a little bit differently along myelinated axons than other areas of the brain. So lots of things use water in the brain, but along the myelinated axons, the water molecules move a little bit different. This is trying to detect that water moving slightly different. So it's mapping that white matter roads of axons, and that's what we can see here. We can see the passive axons because we saw, yeah, the water in this single blue line is moving a little bit differently. So there's clearly some axons here. So this is also very important when you want to, say, detect white matter integrity. So see if there's any axons breaking down, comparing to a couple of years ago. So if you have someone with, say, MS, they're losing myelin and they might be even having damage to axons. So you can look at something like this to see exactly where that's happening. You can also do this for learning, seeing what kinds of axon pathways are being generated in more, because we can generate new axon branches. So at the end of an axon, we've got a couple branches, lots of branches connecting to lots of different neurons. You can see that through a diffusion tensor image, and it does make a really cool picture. But again, what you're doing is you're specifically looking for axon pathways. So that's what each of these lines is representing, is axon pathways. The functional MRI, or fMRI, is looking similarly for the blood oxygen in the brain. So it's not measuring the neuron activity specifically, but it looks at which area of the brain is active, what is using more oxygen from the blood. So similar to the PET, looking at more blood says this area is working. This is a little more specific, looking at that for blood oxygen. And again, it's getting kind of a general space. It's looking at the structures active and working together for about 2 to 3 seconds during a task. It does have high spatial resolution. It's down to about a cubic millimeter in precision, which is pretty small. But remember, that's still a lot of neuron connections happening within that cubic millimeter. So it is low temporal resolution because it has to be recorded over a couple seconds because of the methods that they're doing here. So you get, well, in these couple seconds, this area was active. But you can't say that this cubic millimeter, then this cubic millimeter, then this cubic millimeter, then this cubic millimeter were active in succession. You just see that area was active. So it is a benefit and a low point of the research method. All right, so with this, all of these methods that we've been talking about are correlational methods. It's saying more blood means more activity. So we've got blood blood is higher here in this area, and so activity must be higher. And we're looking at relationships. We're looking at which areas are active as we are recalling a memory. We're looking at which areas are active when we are creating a memory. We are looking at which areas are active when we're planning or when we're making a list, when we are feeling an emotion. But that being said, correlation does not mean causation. So that's a clear piece that I want, a clear thing that I hope you remember from other research methods information. Correlation does not mean causation. Things can be correlated and not causally related, but it does suggest that maybe some of these areas are involved in this process. So here is an example of a positive correlation. Hopefully this is a little bit of review. As this variable goes up, this variable goes up too. So let's say this is, I give a person in an MRI ice cream. Don't think you could actually do that, but I've given them ice cream, and the area of happiness in their brain has more activity. So higher activity, lower activity, more ice cream, less ice cream. I give them 2 scoops of ice cream, they're right here in the middle. I give them 4 scoops of ice cream, they're way up here at the top. So as the ice cream scoops go up, their happiness goes, or their area of their brain with activity goes up. So that suggests more ice cream might be related. So again, this is a relationship. They're related to one another. So here are a couple actual correlations, so from data that I have. So this correlation is one that actually matters here, so 0.33. That's still a fairly low correlation, but it is significant here, and that is a correlation between purpose in life and heroism. So the actual graph of that is right here. You can see there's a lot of outliers. There's a lot of people up here to the top, a lot of people down here at the bottom. It is not nearly as clear as this. We see a clear correlation right here. That is a very clear positive correlation. The positive correlation is 0.89, and that's because this is 4 questions from this 8-question scale. So this is literally half of the questions there. So they better be very highly correlated with one another. So it's half of the same questions as each other. Of course, they should be very correlated. So in reality, oftentimes when we're looking at correlations, they look a lot more like this. And from a casual glance, you might not be able to detect the difference between, say, this one or let's say Let's find the lowest one here. So this one over here, 0.04. So this is an actual relevant correlation. This one is not. This one is basically zero. This one is a weak positive correlation. This is a very strong positive correlation. So, but what we're looking there is relationships. As one variable goes up, the other variable goes up. Is there actually a causal relationship? We don't know from correlations. So that's what all of these imaging methods look at, is correlating behaviors, processes, correlations of. This is why we think the hippocampus is related to memory, because when people are doing remembering, we oftentimes see the hippocampus working. So what do we do about that? Well, to get causal data, again, we have to do experiments. We have to control variables, manipulate a variable, and see changes. So for this simple example, let's look at a simple example of an experiment, something without the brain. So here we have a room. We have a bunch of students who have studied for the midterm exam, and we have a control group that takes the test in a quiet room, and we have an experimental group that takes the test in a room with a brass band playing. So who would you hypothesize would do better? Well, I would hypothesize probably the ones doing the exam in the quiet room. So that would be an example of an experiment. I am manipulating a variable. I'm changing one thing. One group has basically no change in the variables. They're a control group. They have a quiet room versus I'm changing the variable of noise. This group has a loud brass band in the room with them as they take the exam. That is my manipulation. So I can then give them the test, and then I measure their score. So that's the other variable. Now that variable, their test score, did it change? Is this group getting a higher score than this group? I would assume probably. You can make this experiment a little more complicated. You can say, all right, I'm going to have a control group with quiet room, a room with soft music, let's say classical music or something instrumental that is not really distracting, and then a brass band. From that one, which one would you hypothesize is going to do better? I would hypothesize that we would see a similar score between the quiet room and the quiet music room, and then a much lower score from the room with the loud brass band. That's an experiment. We have to manipulate variables. And that is the only way we can see, yep, the noise caused them to get a lower test or to get a lower test score. And so for experiments, this is the only way we can get causal data in the brain as well. And to do that, we have to manipulate brain variables. And so we do that through modulating brain activity. So a lot of this is done in non-human animals, but there are a couple methods to do this in humans as well. So one of these, so this is how we would do the experiment for an animal. We might put electrodes into a brain, and so we will say, all right, I want a similar experiment to this loud and soft room. I am going to have some rats go into a cage and see a cat, and we'll see if they have a fear response. If they run around, they're all scared, they try to get away, that kind of thing. That'll be my control group. My experimental group, I am going to implant some electrodes into the area responsible for calm feelings, or that I assume is responsible for calm feelings in the brain. So in my imaging studies, it looks like this area might be related, have a relationship with being calm. When they're calm, I oftentimes see this area active. Let's put an electrode in there, try to activate it intentionally, manipulate that area. Again, I am trying to modulate or manipulate the brain activity. So I'm going to activate that area and see if this rat behaves differently. So this kind of experiment will give us causal evidence. I can put the rat in there, the rat with no electrodes, and the rat with the electrodes. I can activate that calm feeling center of the rat's brain and see, oh yeah, they are acting. They're not running away. They seem just fine. They're maybe even happy, this kind of thing. That is the only way we can get causal data. So all of these imaging studies are really fantastic for suggesting there might be some causal relationship, but especially when we have so many areas connected. I'm just going to flip back to this. Since so many areas are connected in the brain and activate so quickly with one another, it can be really hard to know, okay, I know this area, and this area are part of this process, or it suggests, the correlation suggests these are probably part of this process. But what's the order of them? We might be able to get more imaging studies that suggest, well, it looks like I see a relationship. There seems to be more activity here, then here, then here. So really at that point, we need to do something like an experiment and say, all right, let's activate what we think is area 1 and see, does it actually produce what we thought? And maybe area 1 was actually area 2. So this is why experiments are so important. And this is why we use so many animal tests, because again, correlations only show us that there's a relationship. It doesn't say that A causes B. This suggests that this area might be related to this process, but to know for sure, we need these experiments. We need to cut out other variables and say, yep, A leads to B. A makes B happen. So that is why experiments are so important. And some of the ways we do that, we already mentioned one for treatment of disorders is transcranial magnetic stimulation. You can use this to intentionally alter a human. You can kind of turn off or on areas of the brain, and you can see if it changes behavior. So do you feel better now? Do this memory task. We're going to try to increase or decrease the memory area of your brain. Try to put these words in alphabetical order, and then we might manipulate one of the areas of the brain, maybe for vision or the frontal lobe for your planning, right? So we can see causally, if I'm manipulating this area, does it change the behavior that I think it might? So again, transcranial magnetic stimulation is used to treat depression as well. That's kind of the same thing. We learned that they can do this because of those types of experiments. We can see that, oh, it looks like these areas of the brain, we see a relationship between their activation when a person is ruminating. So maybe if we activate this other area, it might help the brain not ruminate. Or it might, if we do it in this area, it might interrupt that rumination. And so those kind of causal studies and experiments are really essential to really understanding what the brain is doing all over. All right, another one that we can use, and we do use in humans, is transcranial direct current stimulation. So this uses electrical currents on electrodes on the skull. So rather than the EEG recording information, this is delivering current to the person. It's a low current. It's about like you would experience from a AA battery over an extended period of time. So we have seen that this plus cognitive training can increase math ability, memory, attention, coordination, but it's still a fairly gross interaction, right? This electrode is still pretty big. It's still getting a fairly big area of the brain. It's less and less effective the deeper into the brain that you're looking at, right? So you could put a couple of these and try to get a bigger effect on a specific area, but you're still affecting other areas around it too. Again, better technology in the future will allow us to do this a lot more easily with not as invasive methods. But there are some things that we do in rats. We do these things in rats as well, but there are some things in rats that are a lot more direct at modifying brains activity. One is gene knockout. So this essentially, we are modifying the genetics of rats specifically to remove or make specific genes inactive. And this can help us see the function of that gene in the brain development, in the behavior of the animal. We use this in fruit flies, zebrafish, mice. There's a similar one called gene knock-in, but rather than that, we insert a gene. We put a gene into it, and we'll see some examples of that in just a moment. So this is kind of like what you might expect for the X-Men. So they have different genes that allow them to have different powers. We don't have rats with like laser vision. That would be neat, and pretty terrifying as well. But it's that idea. You're giving them new genes, seeing how it affects things. You're turning off specific genes, seeing how that affects the behavior and the development of the rat themselves. So that's gene knockout and gene knock-in. Another that we use is called brainbow, which is a really cool name, really clever. Sometimes scientific papers and things have really boring names. I named my heroism scale the intended heroic behavior scale because I wasn't creative enough to come up with a better name. But brainbow, that's really fantastic. Essentially, what you're doing here is a gene knock-in. You are transferring a gene from actually the fluorescent jellyfish and coral. So you splice in that gene into a mouse or into a rat, and it can act, you can put this so that their neurons have this genetic trait that with a specific activity, after taking a specific drug, that their neurons will glow. So their axons will glow, the cell bodies will glow, and they can glow different colors. You can insert genes to make muscle cells glow one way, neurons glow another way. The way that we usually talk about it, you're trying to separate individual neurons and their individual axons. So this is actually a picture of a mouse's motor nerves for ear movement. So moving its own ears. That is its motor nerve glowing under its own power and energy because of a genetic mutation, which is really, really neat. Has really helped us see more of how things are connected to one another in rat examples. So here is another example of mouse axons in a brainstem. A lot of density there in the brainstem. And then here are some hippocampus cells, which again, because I double checked earlier, that is the area of the brain responsible for memory. So these are some cell bodies, and you can see these are likely the axons. Right here is probably an axon, and then there's a bunch of axon branches off of that. So with this and with other transgenic, and that's what this means, is it's a transgenic manipulation. You are adding new genes. So you are transferring genes to the animal that weren't there before. So there's a specific type of mouse, like a TET, it's called a TET tag mouse. It's just the little more boring name that the scientists came up with for there. We can actually look at specific neurons that are active during specific things. So some of the things that we've learned, and I've got a paper linked here, is looking at the neurons that are active during making a memory. So a mouse makes a memory. We can look at the neurons that are literally glowing because they have this mutation. We've mutated the mice. So it's made a memory, and that neuron that is part of that memory network is glowing. And we can look at that. It's really cool. So again, this general process is called brainbow to to genetically modify the mouse so that its cells glow. A similar thing is optogenetics. So this is a little bit of the other way, but it's using a lot of the same processes. Again, it's transgenic. You're inserting genes into the mice, and you're modifying their genetics to be light sensitive. So in this case, you can actually activate those neurons that have this gene and make them run by just shining a light. So that's what's happening to this mouse. It has specific neurons in a specific area of its brain that are light sensitive. So when they shine a laser directly into the skull, that activates that area of the mouse's brain. So you put those fiber optic leads, you activate those specific neurons, and you can do a causal experiment to see, okay, can we create this memory? Can we create this decision? Can we treat addiction in a mouse? We have made mice addicted to things and then treated those addictions. Can we make a mouse move in a specific way? So again, we're directly modifying the brain's activity. We're modifying its memory center or its decision-making center. And so we're doing that by first changing their genetics and then shining some lasers into their brain. So you can also do something similar in another way where it's not as specific as this, but you can put direct electrodes to activate areas. This is a lot more specific to get. One, you can put the genes in to both activate those areas of the brain as well as afterwards go in and see which neurons were active during that process. So this is a really great video on how we do this kind of process with rats. It is 5 minutes long, but very worth it to watch. So I'll watch it together with you. Again, if any of my videos ever don't show up, go click on the link yourself and then come back to this video.
Will there ever be a day when at just the push of a button, we can implant or edit specific memories at will?
It's your first trip?
Like in the movies?
The seed that we plant in this man's mind will grow into an idea. This idea will define him. It may come to change everything about him.
Movies like Inception, Total Recall, Eternal Sunshine, of course they're possible. If mice had Hollywood, then it's possible in practice right now.
In fact, here at Columbia University, Christine Denny is one of a handful of neuroscientists who can do just that.
It does seem like science fiction. but we are really doing inception in our lab with turning on and off memories.
It's called optogenetics, a technique so revolutionary it allows us not only to map a specific memory, but manipulate it with lasers. At least in these little guys. These mice might not look so special.
You could not tell my mice apart from a mouse on the street or wherever you would go to a pet store and buy a mouse. They don't look any different.
But they are. These are genetically modified mice that allow Denny to record specific memories and turn them on and off at will. To demonstrate, she starts by putting a mouse in a new environment.
You can see that the mouse is just sitting here in the corner freezing, basically scared of the environment.
That's because it's bright. There's no place to hide. But the goal isn't to frighten mice. She wants to see if she can override this fear by playing back a happy memory she recorded yesterday.
What we did is labeled a positive memory in the brains of these mice.
Yesterday, this same mouse got to explore the kind of place it naturally likes. Dimly lit, full of soft bedding, with a nice place to hide. And while he was scurrying around, Denny recorded the exact neurons that fired when he made a memory of that pleasant place.
The cells that are labeled here in green, when I turn on the laser, those cells will turn on the memory.
But how? How do you record a specific memory? And how do you get brain cells to respond to light? Here's where the sci-fi wizardry comes in.
We genetically engineered mice so that we can permanently label an individual memory.
The key is this mouse's special genome. It's bred to carry a piece of DNA from algae that has the code for a light-sensitive protein. In nature, that protein allows the algae to respond to light. In Denny's mice, it just sits there quietly in the mouse's genome, not doing anything, until...
When you inject a drug right before you expose them to this positive experience,
the drug switches that gene on, telling any brain cells that fire within the hour to install this light-sensitive protein on their surfaces. As the mouse is exploring a pleasant environment, any neurons that fire will leave a footprint of the memory in the mouse's brain. After the drug wears off, only those cells will respond to light. Meaning,
it's basically like a switch. So what you can then do is use a laser to control these cells.
These tiny fiber optics can shine light directly into the mouse's brain.
What we're going to try to do now is to turn on these cells that we've labeled with a positive memory.
Right now, the mouse is scared. But if Denny is right, the laser should activate the exact same neurons that fired when the mouse was making a happy memory, effectively causing it to relive that positive experience.
Okay, so watch now what happens when I'm going to turn this laser on. You can see that the animal is actually smelling, grooming himself, which is a sign that he feels safe.
But turn the laser off,
and you can see that the animal is resuming its behavior of freezing in the corner.
Denny can now trigger this memory at will. On, off.
I think the first time we did it, we didn't believe it. But when you see inside of the brains of these mice, and then to think that you're only manipulating those cells and changing the behavioral output of the animal, that's, yeah, science fiction.
This is potentially one of the most important new developments in memory research because it suggests a level and precision of control over memory that we've really never seen before.
A degree of precision many scientists think we might have over our memories someday.
I think that it's a matter of when this happens, not a matter of if it'll happen in people.
Which raises the question, if by a flick of a switch we could edit that first kiss, or erase that argument with a spouse, would we want to?
A pretty crazy example of all of those research methods that we've been talking about being put into place to literally, one, genetically modify a mouse using these transgenic methods. So you now have a genetically modified mouse. It can now, you could record and see where you give them a drug, have them go through an experience. Then afterwards, you can look and see what neurons were active during that process, as well as you can now manipulate those same neurons. Again, this is a experience that takes a long time. It's not super temporally specific. You couldn't make that mouse remember, say, like this color versus that color yet. But it is a lot more specific control than we've ever had before over these kinds of memories. So it is really crazy science fiction kind of things. Very interesting to see what we are learning from the brain through these causal experiments. And that's the main idea and main point I'm bringing this up for, is that these are the methods that we're using to, one, look at correlations in brain activity. So remember, correlations are those relationships. We can look at an EEG or we can look at an fMRI and say, okay, these are the areas that look like they're active while a person is doing these processes. But that just gives us a relationship. We don't know for sure that those areas are causing this kind of thing. But by doing these types of experiments, we can see by activating these neurons in the mice, it is making their behavior change. It looks like behavior that is not fearful behavior. So whether you could say that's exactly that it's the memory, or you have also connected to emotion pieces, really those emotion components of the brain are part of your memory. So those neurons that were active and made you feel happy during the memory, you rerun those. You re-go through those same neurons when you think of that memory and feel happy too. Um, Spoiler alert for later on, but that's also why sometimes memories change. Let's say you had a bad breakup or a bad argument with that person. Now, when you think of that happy memory, it might be tinged with a little bit of unhappiness too. So your memory is now shifted or moved a little bit. You've got new neurons in there. So in humans, we use transcranial magnetic stimulation. We can do implanting electrodes. Remember that implanting of electrodes, it's only done if someone's already going through brain surgery. In mice, though, we can do these. We can also do gene knockout, gene knock-in, transgenic manipulation so that they are born with these genetic mutations, and using optogenetics to, one, record which specific neurons were active and activate specific neurons. And it's through these kinds of causal methods, experiments, that we can really see what parts of the brain are active. So while this was a lot of new stuff and a lot of really interesting things, well, I find it very interesting for how we can do these kind of brain studies, whether it's imaging, whether it's causal studies. The main idea here is to give you a sense right now of the kinds of ways that we do study the brain and how we do these research methods. So this is the model I kind of use in my research. I find socially significant problems, things that maybe need to be solved. I do research to help solve the problem, but with that, I get out and work with people in the real world, especially those of you in behavioral health. You know that you need collaboration with communities, with practitioners, with the people themselves. And you use that, you work with what you have, the theory that you have, the research methods that you have, and then based on your findings, you give that information to people, whether it's the people themselves, whether it's to evaluators, policymakers for things like changing high school start times, other researchers. And the idea is to work on these problems, to see what we can do, what technologies we can develop, the ways that we can measure things to be better, to eventually solve problems. That's the idea behind brain research, is to use these kinds of things to have a much better understanding of how does memory work? How does emotion work? How can we encourage plasticity of the brain? What are technologies what we can use or medicines that we can use to change the chemistry of the brain to make people more plastic in their brains or recover quicker from something like trauma or depression? So hopefully you see some of these connections and you can come back to some of these slides later on in the class as we talk about more research and findings from these research methods. But that is all for Chapter 1. And remember, as you go throughout this class, double check your schedule in your syllabus to make sure what chapter to go to next. Sometimes we'll go right in line, but depending on which quarter that you're taking this class, depending on what we're doing, we might be jumping around to different chapters. So double check your schedule, and welcome to biological psychology and neurobiology in the behavioral healthcare program.